Hello and welcome to my exercise application video number 6, I believe. We just finished up in class today talking about all of our content for exam number 1. So I have a lot of content to cover in these next couple videos, so I'm just going to kind of focus on one thing at a time. And today I'm going to talk about what actually happens inside of a muscle cell once an action potential is transmitted. So I've talked about this. Our nervous system is essentially an electrical system. Specifically with action potentials, it's based on the movement of ions, which are charged particles, and thus they can carry electricity and charge. So it's pretty cool to think of it that way. But what happens is, so again, that signal, I've talked about how that happens regarding the sodium and potassium movements, but an action potential is propagated down the axon and then it goes to the synapse. And so our synapses have these little synaptic vesicles that are just holders for different neurotransmitters and hormones. So typically with muscular contraction, we're talking about norepinephrine, epinephrine, acetylcholine. So we'll just say we're talking about epinephrine for this video's sake. So your brain decides to do a movement. You consciously decide to do a movement, and then your brain sends an action potential down your motor neurons. And so then that signal goes down in acetylcholine in its synaptic vesicles. It binds that synaptic vesicle to the presynaptic membrane, and via exocytosis, it just kind of spits out all of that acetylcholine into the synaptic cleft or the neuromuscular junction, whichever you prefer. And so the receptors then on that motor end plate grab that and they experience a depolarization because of, again, the changes of ions. And so then transverse tubules inside the muscle cells actually help carry that charge throughout the muscle cell, deep into the muscle cell, and they do it quickly. And they also then are beside the sarcoplasmic reticulum, which wraps around our myofibrils. And this sarcoplasmic reticulum has voltage feet or calcium feet, and once they experience the charge from the transverse tubules, they release calcium ions. And what these calcium ions do, our muscular contraction is based on something called the sliding filament model. So essentially we have myofibrils, which are contractile units of our cells, and they're made up of sarcomeres, which range from Z disc to Z disc. And between those Z discs, we have thick filaments, which is our myosin, and thin filaments, which is our actin. And these are just part of the contractile components of muscle cell, which is very important for us to move our bodies properly. But our myosin has troponin that binds to it and tropomyosin. So tropomyosin wraps around the actual cell, and then troponin sits on top of each tropomyosin. So when calcium from the sarcoplasmic reticulum is released, it binds A to that troponin, unlocks it, and then tropomyosin acts as a deadbolt and it shifts. So key, calcium, lock, deadbolt. It shifts off. The myosin heads are then able to grab onto the actin and contract them inwards. So during contraction, muscles move in towards their muscle belly on both sides. And so what happens is that requires ATP to do that. And so that ATP is kind of recycled. So an ADP is released off, you get 1/2 crank of the power stroke for the sliding filament model. Then you release an inorganic phosphate, boom, you have a full stroke. But to actually release that myosin head from the actin, you have to have another ATP come in, which is why our bioenergetics are so important. Also with the sodium potassium pump, which is involved in this neural transmission. And so yeah, the myosin heads have something called myosin ATPase on them, and however quickly that myosin ATPase determines if something is a slow twitch or a fast twitch fiber. Fast twitch fibers have faster myosin ATPase, and they're able to quickly recycle through ATP, which allows for them to contract quickly in a short amount of time. Whereas slower twitch fibers, their myosin ATPase is slower, so they're not able to recycle as quickly, but that is more sustainable for them, and that's why they can endure for longer periods of time. Today, I was using more fast twitch fibers, which I will talk about in another video, but again, the signal goes down to synapse, synaptic vesicles, exocytosis. It's picked up by the end plate, which is depolarized, and then that goes through transverse tubules, sarcoplasmic reticulum, releases calcium, and then that unlocks troponin, tropomyosin. Myosin grabs actin. That ATP is hydrolyzed, and then we recycle through, and that is how our muscular contractions work once we get actually inside the muscle.